happy to be here at that very nice meeting, and I would first like to thank the organizers for the opportunity to present this talk here. So I have to say I'll switch a bit gears here, because I will not speak about creatine supplementation. There is one slide just at the end. I will not speak about creatine transporter cancer and a lot of other issues that have been already presented here during this meeting. I will present some concepts and a model that we have proposed in the context of a new role of the creatine and creatine kinase system in heat generation, mainly in adipose tissue. Okay, so we are just starting a project, so I will neither present data from that project yet, but I think it's important to have a view on that new mechanistic task of creatine kinase and creatine, and to keep that in focus, because as you will see, there have been about 20 very high-profile papers, and there is Cell, Nature Metabolism, Cell Metabolism, during the last 10 years on that topic. And there is quite some controversy also in that field, very harsh sometimes. And so there are opportunities, okay, but still the field is just developing, okay. So I would like I would like to first explain a little bit, because I think most of you are not very interested in adipocyte thermogenesis. What is that? What is thermogenesis in a living organism? And then why there is this increasing interest, why we have these 20 papers in Cell and Nature. And then, as you have seen already, futile cycling. What's futile cycling? Maybe you heard during your studies once, and I will try to explain. And then giving some examples of that. And then in the end, giving you some ideas on what's considered today as a creatine futile cycle that can generate heat, okay? And what's the pros and cons on that. Okay, so my first contact with that phenomenon was, in fact, over 20 years ago in the lab of Theo Wallimann, where we studied a double knockout mouse for mitochondrial CK and BCK, and I tried without success to breed these animals, okay? And one day I looked at them closely, and they were sitting in the corner of the cage and shivering like crazy. And I thought, okay, they just feel not good, okay, so let's buy some heating pads. I put these heating pads in the cages, and they were very, very happy with that and breeded. And we went on with our other project, and I forgot a little bit about this problem in thermoregulation. But in fact, it means that they require thermoneutral conditions to reproduce. And be aware that the thermoneutral condition for rodents is for 28 to 30 degrees, okay? So you have to put them in this temperature range to be happy without being reliant on a supplemental heat generation in their body. At that time, we didn't went further with that. These were mice generated by the group of Beveringer, and they went a little bit further, tried to really analyze why they have this problem in thermoregulation, and data were not very conclusive. So their conclusion was, finally, it's a central problem rather at the level of the brain and the adrenergic stimulation of the brown fat, but they didn't really could nail down the mechanism. And as you see, this is 20 years ago. So things changed again 10 years ago, when this Cell paper was published by the group of Bruce Spiegelman at Harvard, and they had quite some convincing data, totally independent of our studies at that time, that there is an involvement of creatine in a creatine-driven substrate cycle that enhances energy expenditure and thermogenesis, at that time in beige fat. I will explain that, but also happening in brown fat. And there were some issues that were not so quite clear. At the same time, there was a group in Zurich by Christian Wolfram who published data that went into a similar direction, meaning that if you expose animals to cold, they will upregulate creatine kinase metabolism-related genes, different isoforms, and in these fat cells, there are probably all 4 different isoforms expressed. So BCK, MCK, the 2 mitochondrial. They will upregulate creatine biosynthesis enzymes. They will upregulate creatine transporters. So for sure, there is a role of creatine in thermogenesis. Okay, so that's just the introduction. I would like to go now to the point, okay, thermogenesis, what's that? Why we need that? Thermogenesis, first, to come to that, we have to be aware that if we go for transformation of energy from food to work, there is not only the work that will be the final result. If we look further in thermodynamics, every change in internal energy of the system will partly be a change in the work done on the environment and on the heat that is exchanged with the environment. So every chemical reaction, whatever you take, will, if you want, do a lot of work, but also produce some heat. And you can shift any reaction to a situation where it will do less work, but then with the same change in energy will do a lot of heat, okay? So you have the choice, or nature has the choice to shift reactions to be very efficient, let's say, doing a lot of work, or being very inefficient, generating a lot of heat, okay? So that means that for your energy intake, going into metabolism, of course, you will partially have the possibility to stock that energy, but then you want to do work with the energy that is transformed here by metabolism. And, well, there will be always heat, and together with the work that is done. Meaning that as an inevitable result of metabolic activity, all living organisms are thermogenic at different degrees. And the fact that you can generate heat in an adaptive way allowed finally the evolution of homeothermic animals or hibernating animals, which is the most extreme situation when animals have to survive during harsh winter without moving anything, just sleeping. Okay, so there is always heat produced, but it's different kinds of heat. So that's what is called thermogenesis. It's different kinds of heat that can be produced in an organism. So you have this obligatory expenditure, which is associated with all your metabolic activities, okay? Because your biochemical reactions are never 100% efficient. Then there is some heat generation that is linked to your physical activity. Okay, so that's mainly your muscles, maybe your heartbeat that accelerates. One may even include brain activity above, your sleep basal activity. And then there is adaptive thermogenesis that has developed. And this adaptive thermogenesis is variable, is regulated by the brain, responds to temperature, and likely, although there is some discussion on your diet, when you eat, you may induce thermogenesis above the basal levels of an accelerated metabolism. And this adaptive thermogenesis mainly occurs in the mitochondria of adipose tissue and in skeletal muscle, and maybe in some other sites, okay? And what we are speaking here about, where creatine is involved, is in this adaptive thermogenesis. Okay, it happens, as we have seen, mainly in the adipose tissue. There is brown and beige adipose tissue, and that is also why it now became so fashionable to work on that, because it was a long time not so clear that humans still have brown and beige adipose tissue was long meant that babies have some brown fat, but then somehow adults, they don't. And now it's known, if you look at these PET scans, there is quite some brown adipose tissue, especially here in this subclavian region, but also in this paravertebral region. And there is also some so-called beige adipose tissue that is more in the abdominal region. So what's that? In fact, from different pre-adipocytes, you get a differentiation into this adipose tissue. So the white adipose tissue is really there to stock energy, okay, to stock lipids. And here, you virtually don't have mitochondria. Okay, that's really for storage. On the other extreme here, you have the brown adipocytes in the brown adipose tissue. As you see, they have some relatively small lipid droplets, and they have a lot of mitochondria, okay? And they really evolved to generate heat. You will see by which mechanism. And so that was meant long time as the only tissue that can generate heat. But in fact, there is a possibility of white adipose tissues to develop a little bit into the direction of brown adipocytes. As you see, you have fragmentation of the lipid droplets, you have a proliferation of mitochondria, and so they will also be able to generate heat. Okay, and that differentiation here happens under, the exposure to cold. So really you have to be cold exposed for some time to go into that kind of differentiation. And it's not all the white fat, it's really part of the white fat that can go to this browning procedure. Okay, so evolution has really established a specific organ that can generate heat very well. So how does that function? No, first, okay, that was the first point, why it became very, very fashionable to work on that. And the second point, why it became very fashionable is, of course, that this holds a promise to treat obesity, okay? If you are able just to burn your fat in your brown or beige adipocytes, you don't care about all the rest, you can eat what you want, and to somehow trigger then your thermogenesis, and you lose weight, okay? Or even in winter times, you may, do some economy on your heating because you're always well generating your own heat. And a second aspect, of course, is also that this kind of thermogenesis may not only be triggered by cold, but also by your diet. And this diet-induced thermogenesis, as you see here in this title of this article, is also somehow disputed. Still, how much it is and under which conditions exactly it is triggered. But it is likely a possibility that we have to keep in mind, okay? So that is a big promise that thermogenesis in brown and beige fat can act against the big obesity crisis. So, how the heat is produced, okay? So heat is produced in short term, and that's your personal experience, I suppose, by shivering, okay? Muscle shivering, that's the immediate response. But then there is an adaptive response in the long term, and this is then the non-shivering response, and this relies on so-called futile cycles, okay? There is a futile cycle in mitochondria that depends on proton futile cycle by a protein called UCP1, uncoupling protein 1. And there are futile cycles that do not depend on this UCP1, but are ATP dependent, like calcium futile cycle, lipolysis, lipid synthesis futile cycle, and creatine futile cycling. And this is just an overview, I'll explain. Don't be afraid. But what's futile cycling, okay? Futile cycling means you use energy to get some sort of activated product, and then you don't do work, okay? You just dissipate all the energy here into heat, okay? For example, you pump an ion across a membrane, but you don't establish a gradient because there is a hole in the membrane, and the ion just goes back. Or you phosphorylate a compound and immediately do hydrolyze without doing work. So all that means that, as I said, you entirely switch off the fact that you could use energy as work, and you dissipate all the energy change, for example, of ATP hydrolysis, into heat. Okay. So that's the principle of futile cycling. So how that is realized in brown fat, and that here, left hand, this is the traditional principle of thermogenesis in brown fat. This is also called uncoupling. That's why it calls uncoupling protein 1 here. And this is nothing else as a hole in the membrane for protons. Okay? You have here the respiratory chain, the reduced substrates coming in, proton pumping in complex 1, 3, 4. And normally this gradient, this proton gradient, is then used by an ATP synthesis to generate ATP. But instead, you have here UCP1, and this is like a short circuit in your electric installation, okay? The protons just return, doing nothing else. No gradient is established anymore. And as you know at home, if you have a short circuit in your electricity system, that may heat up, that may even provoke a fire in your house, be careful. Just happened to a colleague of mine. Here, you produce a lot of heat just by this proton cycling. And so that's the main mechanism. And all other mechanisms that we will discuss have to compare with that, okay? How efficient they are, how much they can contribute. More recently, as just a few years ago, it was shown that also the well-known translocase of adenine nucleotides, ANT or AAC, is able, if it is stimulated by high amounts of fatty acids, to uncouple, that means to transfer protons. And one aspect also important for that kind of futile cycle, it is regulated. It is very important, of course, for adaptive thermogenesis that you can switch it on and switch it off. And here, it's the mobilized fatty acids, normally in the lipid droplets here, already degraded to fatty acids. They are activating UCP1. They are activating ACC. Under normal conditions, if there are no fatty acids, if there are a lot of nucleotides around, UCP1 is inactive, UCP1 is not uncoupling. So you have a possibility to switch on and off. Okay, so now what's about, and you can produce here a lot of heat, of course. So now what happens with these other futile cycles? Okay, these futile cycles now are ATP dependent. So already they require that there is no uncoupling by UCP1 or very few. So there could be some production of ATP by ATP synthase from the proton gradient. Then you have sufficient ATP. And then you can, for example, have a futile cycle in the endoplasmic reticulum. In the endoplasmic reticulum, you have this highly energy-dependent calcium pump called circa. Theo Wallimann already mentioned in his talk. And this circa will pump calcium into the ER by using ATP and establishing a very, very strong gradient, 2-3 orders of magnitude. But of course, if you have here a channel that is open, this is a ryanodine receptor that is normally releasing calcium, for example, during muscle contraction and other situations. If this one is open, there will be pumping, but no gradient generated. Calcium will immediately leave mitochondria and be pumped and leave and pump, and nothing changes, in fact, okay? No gradient, so heat generation. And then there are more metabolically linked futile cycles. There is a lipolysis re-esterification cycle in the cytosol, potentially. We speak here always of adipose tissue, but that could also happen in muscle, where you normally generate acylglycerols from free fatty acids using ATP, and then you have immediately a lipolysis, and nothing will enrich, okay? And you will just turn in a circle and generate heat. And finally, we have what we want to see in the context of that conference. That is the creatine futile cycle. And you see, yeah, well, we have already seen that before, like in the UCP1, you need here a gradient of protons, okay. In this case, as I mentioned, is used to generate ATP by ATP synthase, okay, so far so good. So now we have ATP, which is exported here by the transporter outside the matrix space in mitochondria, okay. There is sitting the creatine kinase, the mitochondrial creatine kinase, transphosphorylating ATP to phosphocreatine, still good. But then this phosphocreatine doesn't leave the mitochondria according to that model, because there is a phosphocreatine phosphatase, okay, which immediately hydrolyzes phosphocreatine to regenerate creatine. And then you have here a circuit that is constantly turning without doing work. The energy in phosphocreatine is lost and dissipated as heat. So in that model, the creatine phosphocreatine cycles inside mitochondria, the outer mitochondrial membrane would be up here. And of course, that cycle would generate also a lot of heat, but will of course depend how much ATP the cell can produce. So some details on this cycle, okay? Here is creatine kinase, phosphocreatine is generated, will not leave mitochondria. The phosphatase sitting here, directly regenerating creatine. Now for the sake of time, I will not go through all the figures of the 20 high-profile papers in support of that shuttle, okay? I will just summarize. There have been some changes in the model because the first publication claimed that it's a mitochondrial CK and a phosphatase called phospho-1, which is a phospholipid phosphatase, but didn't turn out to be the right one. So there were 2 publications in Nature in 2021 stating that it is an intra-mitochondrial BCK, so the cytosolic enzyme being in mitochondria for the creatine kinase. And there is an intramitochondrial TNAP, which is a tissue non-specific alkaline phosphatase, which is known as an extracellular enzyme, for example, involved in bone mineralization, okay, also in mitochondria. What's the support? The support is, as I mentioned before, cold adaptation. All the creatine kinase-related enzymes are upregulated. And this has been shown by the Spiegelman group, has been shown by the Wolfram group, has been shown by others. Second, if you knock out specifically in adipose tissue CK or the phosphatase, let's say here specifically BCK and TNAP, you impair the UCP1 independent thermogenesis that can be observed. Quite clear. What's the evidence on a cellular molecular level? There it becomes more difficult. The main argument of the Spiegelman group here is that if they give external creatine, okay, they will get more respiration of mitochondria that could be calculated by the amount of creatine. Because a certain amount of creatine will, of course, by this coupling to creatine kinase, generate a certain amount of ADP, which would be good for a certain amount of respiration in the respiratory chain in mitochondria. If you get more than that calculated amount, this means that there is some cycling here going on that constantly where it generates ADP because the phosphocreatine here is not accumulating. It's immediately hydrolyzed to give creatine, which could again, take an ATP and transphosphorylate and generate an ADP. And it's the ADP that will stimulate respiration. So there is a problem in stoichiometry, and that's the main argument in these papers. What's the problems with the model? Well, as you already see here, you have to do some big claims, okay? You have to claim that BCK is moving here into mitochondria. The evidence for that is not really very unambiguous. And it's true for both of these proteins that have never been shown really to be localized here within mitochondria. And then there is a problem of specificity and activity. While BCK is a highly active enzyme, high turnover number, very specific for creatine, exclusive substrate, the phosphatase that is now examined here, TNAP, is finally hydrolyzing a lot of different phosphor compounds, not only creatine, also ATP, ADP, others. And its activity is far, far below what we see for creatine kinase. Here they isolate phosphocreatine with an extract from adipocyte mitochondria from cold acclimated mice. They wait 6 hours to see some decline here in the phosphocreatine peak. Okay? If you want to have a futile cycle going on with creatine kinase within milliseconds generating phosphocreatine, you need a phosphatase that in milliseconds will hydrolyze phosphocreatine to go back to creatine. So one has really to postulate that there are some huge activation steps involved or co-localizations, interactions, whatever. You could do, of course, but it needs really these strong changes that you have to postulate. And finally, if we say, okay, there is a futile cycle, it is also the question how much it can contribute. Here, these are respiratory curves, controls in presence of creatine, and they also use very low concentrations of creatine, which where creatine kinase is hardly active. And you see here in these conditions here with differentiated pre-adipocytes, the addition of creatine here has very, very few effect, yeah? And maybe even within the margin of errors. And if you compare that here, with warm and cold adapted animals using UCP1-dependent thermogenesis, you have an increase of 2 to 3-fold in respiration. So UCP1 is extremely efficient. The futile cycle by creatine, we don't know yet, how much that could contribute in total to adipocyte thermogenesis. Okay, so as you see, there are pros, there are cons. I will be careful here, but as you see, we have with Theo written an opinion paper some years ago, which took some effort to get that published. So we were at least the first, but as you see, there is quite a bunch of papers since that people say, okay, there is a lot of truth in that, but is it really working as it has been published so far? And this includes a paper in Nature Metabolism. That's good to also have these journals accepting that there is a dispute, written by Dave Nichols, who is a real specialist in mitochondrial physiology and has worked himself on UCP1 thermogenesis. And there is a very recent discussion paper published in Cell Metabolism, where there is a clear point-counterpoint argumentation by Barbara Cannon and Jan Nedergaard, who make a lot of doubts on creatine futile cycling, and Lawrence Kazak, who was part of the Spiegelman group, who defends the model that he has published. So, of course, as you see, there are lots of questions within that field. And what we have, in our opinion paper, proposed is a model that may reconcile at least some points that are difficult to understand at the moment. And this proposal is based on the shuttle that already Theo Wallimann showed you in his talk, which is the classical connection between energy or ATP generating sites here in mitochondria and with glycolysis, and the ATP using sites, the ATPases that then do the work. So that's the classical shuttle. Now then we can use here as an ATPase, we could use the circa, you know, the calcium pump, that, as I showed you, is known to be involved in a futile cycle. That is quite well accepted. Futile cycle of calcium within the sarcoplasmic reticulum or the endoplasmic reticulum. And it's known that there are different transporters or channels here in the ER membrane, like the ryanodine receptor, the IP3 receptor, that are able to switch in a mode where they constantly are open and then allow this type of cycling of calcium without establishing a gradient. So there is cycling, there is use of ATP, and there is only dissipation of heat. And another advantage, in fact, of this cycle is it is known how it is regulated up to the molecular detail. So there is, you know, sympathetic nerve activation, then the adrenergic system that triggers IP3 signaling, CMP signaling, that then trigger the opening here, and even, and that also has been published already, uncoupling of circa B to B, meaning that it hydrolyzes ATP and even does not transport calcium. So again, a sort of futile stuff. There is ATP hydrolysis, there is no transport, so again, heat generation. So there are lots of advantages of this calcium cycling in the endoplasmic reticulum here. And we think that could explain, for example, why it is the BCK that is really important. Because I think I forgot that a major argument, of course, in the papers of the Spiegelman groups is their knockout models. So they have knockout models, tissue-specific, meaning adipose tissue-specific for BCK, for TNAP, and they show impaired thermogenesis. That's a clear fact. So you have to explain how BCK can be involved in futile cycling. And here it is known that BCK can interact with circa. And so in that case, you don't need a BCK that is intramitochondrial. You have the BCK in the cytosol that interacts with circa and is here at the receiving end of this of our creatine shuttle. And so you don't need neither a phosphatase. That is relatively difficult to imagine how a phosphocreatine phosphatase is intracellular and especially in mitochondria, because it is a potential danger for high energy compounds like ATP or phosphocreatine. So that model here could at least resolve some of the issues, and we are currently working on both types of models and look what we can find during cold exposure. So what we can finally say about creatine and potential creatine supplementation in that context. I think that is most of you are interested in that. So in fact, there are some data, at least, okay? There are some data with animal models, okay, where they have depleted, in fact, the adipose tissue by knocking down GATM or AGAT, the rate-limiting enzyme of creatine synthesis, okay? And they found that, okay, these mice become obese on a high-fat diet. Okay, the cold and adrenergic mediated thermogenesis is impaired, and dietary creatine can rescue at least the adrenergic thermogenesis. So as you see, it will not rescue all the phenotype, but partially there was some effect of creatine in restoring a phenotype that has been induced by depleting creatine, specifically in adipose tissue. And I think based on that finding, the Spiegelman group, together in collaboration with the group in the Netherlands, started an interventional study with adult vegetarians in a double-blind, randomized, placebo-controlled crossover trial, very well designed. 14 young people, well, it's not a big group, and they didn't find any effect on thermogenesis. So they looked on thermogenesis induced by the diet, so with the meal. They looked at thermogenesis induced by 4-degree exposure with a very sophisticated protocol with a water-filled suit and exactly controlled temperatures, very well designed. Well, I think that's why it ended up in Nature Metabolism, but didn't provide evidence for an effect of creatine supplementation. Also, clearly, they had more phosphocreatine, they had more creatine, and they had a better mitochondrial function. So global parameters of creatine supplementation were there. Okay, so you see also relatively complex, yeah, if creatine can, or creatine supplementation can boost such futile cycles. So finally, what we can learn for the moment of all these data. Creatine kinase play a role in brown and beige adipocyte thermogenesis. I think that is clear, that's a fact, okay? And this probably functions by futile cycles. But the exact molecular mechanism, as I showed you, is not yet clear. Is it really intramitochondrial? Maybe there is an extramitochondrial loop. Maybe you need a phosphatase, you don't need a phosphatase. Not really clear. The contribution of such ATP-dependent thermogenesis in general may be limited because it depends on how much ATP is left after UCP1 is already functioning, okay, and uncoupling partially the mitochondria. So these cells may not have such a lot of ATP to support these futile cycles. Now, there's a recent paper showing there may be different kinds of brown adipocytes. One more dependent on UCP1, others more dependent on ATP and ATP-dependent futile cycles. So that would be, of course, an option. But of course, the bottom line is that further research is needed, particularly also regarding potential effects of creatine supplementation, because despite all these high-profile papers that we have seen, for a lot of people in the field, it's still not quite clear how exactly this cycle functions. Okay, and with that, I thank for your attention. I would just like to present our lab crew and the people involved in the project that we just started only 2 months ago, Marco Schada-Tokarska, Laurence Kay, and Oran Leroulet, our postdoc who is present here in the audience, our funding. And yeah, thanks for your attention, and you can always write me if you have any question.